Hello everyone, myself harapriya Sahoo, HOD Department of Botany, and I am going to describe about the structure and life cycle of Marchantia. First of all, we know about the Marchantia. Marchantia, as we know, is a bryophytes are the amphibians of the plant kingdom, and we will go to the systematic position or its classification. Marchantia belongs to the class Hepaticopsida and order Marchantiales and family Marchantiaceae, and it belongs to genus Marchantia. Its general characteristics, Marchantia is as you know that though bryophytes are the amphibians of the plant kingdom. So, Marchantia plant body is a thalloid, and that means it is a flat leaf like structure, and it possesses the dorsal view and the or we can know that the upper side and lower side view. First, external surface of the thallus, it shows prominent midrib at the apex of which there is a growing point situated, and that growing point is dichotomously branched. Dichotomously branched means the apex is divided into two parts, and the two parts are the growing parts. So, that is called as the dichotomously branched. On the upper surface of the thallus, there are presence of the cup like structure which is called as the gamma cup. These are the reproductive bodies of the thallus. Then inside the gamma cup there are a number of gamma stalked bodies, and which helps or which are meant for the asexual reproduction. In the thallus of the dorsal thallus of the Marchantia, there is the presence of the stalked reproductive organs like antheriophore and archegoniophore. This is the thallus structure, and the lower side of the thallus bears scales and rhizoids. The rhizoids are of two types. One is the tuberculated rhizoids, and another one is the smooth walled rhizoids. Smooth walled rhizoids and tuberculated rhizoids are different because smooth walled rhizoids possesses smooth surface, whereas the tuberculated rhizoids contains inner wall of the tuberculated rhizoids contains peg like infoldings, and that is why they named the tuberculated rhizoids. It is the ventral view having the rhizoid scales, and scales are arranged in 2 to 4 rows on each side of the midrib. Midrib is prominent on the upper surface of the thallus. Then internal structure of the thallus. The thallus of the Marchantia is differentiated into two zones. The upper zone is the assimilatory zone, or we can say it as the photosynthetic zone, whereas the lower zone is the storage zone. The upper zone possesses a discontinuous midrib, discontinuous epidermis which is called as the upper epidermis, and it is ruptured or its discontinuity is due to the presence of the air pores. Inside the air pores there is the presence of branched green filamentous structure which is called as the photosynthetic filament. Each photosynthetic filament chamber is separated by a partition wall, that is called as the photosynthetic wall or partition wall, and the photosynthetic chamber is open towards outer side by the pores, and this is the upper epidermis. And all the cells of the photosynthetic zone are green in color, and there are the chlorenchyma cells. Then the storage zone, this is the storage zone. The storage zone cells are compactly arranged without any intercellular spaces. Storage zone is being limited by the lower epidermis, and the lower epidermis contains the smooth walled and tuberculated rhizoids. The storage zone contains the storage product as the starch as the storage products. This is the another view of the thallus of the Marchantia. This is the internal view. Next come to the asexual reproduction, or the reproduction in case of Marchantia takes place by two methods. One is the sexual reproduction and another one is the asexual reproduction, and this is the asexual reproduction. Asexual reproduction takes place by fragmentation, adventitious branches and gamete production. Fragmentation means when the thallus grows, the lower portion of the thallus becomes older, and the older cells dies, and continuous death and decay of the older cells reaches a portion, that is the apical portion, and that apical portion is divided and separated into two parts, that is called dichotomous branching, and each branching is capable of producing a mature Marchantia thallus. Then adventitious branches which develops on any part of the thallus, that is the extra branches which develops from the any part of the thallus or from the thalloid or flat leaf like body, and on separation from the mother plant they develop into a new plants. And third one is the bigamy. Gammae are produced on the upper side of the thallus inside the gamma cup. The gammae are biconvex stalked body and they contains the rhizoidal cells, the chlorophyll containing cells. The rhizoidal cells on separation from the lobes they develops into rhizoids, and the chlorophyll containing cells next develops to the thalloid body. This is the gamma cup and these are the gamma. Next is sexual reproduction. Marchantia is oogamous in nature. Oogamous means the male and female reproductive organs are morphological and physiologically dissimilar. All the species are dioecious. Sex organs are antheridia, male organ, archegonia, female organ, antheridia born on antheridia 4 and archegonia born on archegonia 4. So, sex organs appear once a year in April and May. Antheridia. Antheridia 4 are differentiated into stalk and the disc. The disc is somehow 8 lobe representing a single branch of the thallus. It resembles thallus and divided into air chambers alternating with antheridial chambers. Antheridia are born inside antheridial chambers. They are arranged in 8 rows on dorsal surface of the thallus representing a single branch of the thallus. These are the antheridia 4, structure of the antheridia 4, that is stalked bodies. This is the structure of the antheridial disc and these are the antheridial present inside each antheridial chamber. Then mature antheridium. A mature antheridium is divided into a stalk and a body. A body has a single layer jacket layer or antheridial wall that encloses a large number of androcytes which gradually metamorphoses into antherozoids. The antheridia dehisce or rupture due in the presence of water, then sterile jacket layer of the antheridia absorb water and the antherozoids release from the antheridia and in a mass and swim in water film. This is archegonia and this one is the archegonia 4. So, archegonia 4 is also called as the carpocephalum. This is differentiated into stalk and a disc. Stalk is short and elongates after fertilization. It is also 8 lobe directed from the center. Archegonia develops on upper surface of the disc, but after fertilization gradually the disc elongates and the archegonia hangs towards the lower side, that is inverted. Each row represents single lobe of thallus and bears 12 to 18 archegonia. These are the archegonia 4s. Archegonia. After fertilization, the stalk elongates, central portion of the disc bulges after pushing archegonia at the periphery. It results in complete downward movement of the archegonia. It shifts youngest archegonia to reach near stalk and oldest unfertilized towards the periphery. So, intercalary growth occurs between fertile lobes which results in formation of radially elongated green outgrowths called rays. That means before the development of fertilization the archegonia are present on the upper surface of the disc, but as fertilization takes place the stalk enlarges or elongates and pushes the disc towards the lower side of the archegonia 4 and they come towards the stalk region. This is the structure of the archegonia 4 and this is the archegonia 4 where it elongates towards the lower side and these are the archegonia. Internally the archegonia is divided into two parts. The upper narrow portion is called as the neck and the lower swollen portion is called as the venter. Venter contains one ventral canal cell and another one is the large cell, that is the egg cell, whereas the neck contains neck canal cell and it is covered by neck cells. Then fertilization of the Marchantia occurs in the presence of water. Male and female grows close to each other. The antherozoids swims or moves towards the archegoniophore, and the antherozoids fuses with the haploid egg cell and form a diploid zygote. From the formation of the zygote, the sporophytic phase starts and the gametophytic phase overs. This is the archegoniophore structure, and then the sporophytic phase. As the zygote is produced, the zygote is then converted or developed into the sporophyte. Here the sporophyte is diploid in nature. The sporophyte in Marchantia is divided into three parts: foot, seta and capsule. The foot is parenchymatous and it is attached to the archegoniophore or thallus of the archegoniophore, whereas seta is the middle cell, an elongated cell, and it is connected with the interconnecting link with the foot and the capsule, and the capsule is the bulbous region, circular in, spherical in structure, covered by a layer, that is capsule wall, and it encloses a number of cells that is called as the spore mother cells. These spore mother cells are diploid. They undergo reduction division and produces the haploid spores, and in accordance with the spores there are also some cells which are called as the elater cells. The elaters are elongated cells which spirally elongated cells which helps in the dispersal of spores. The capsule is protected by two layers of protective covering, that is the perigynium and calyptra. So, the capsule encloses the spores. When the capsule matures, the spores are released by the dehiscence of the capsule. Dehiscence of the capsule means there will be slit in the capsule wall from which the spores will be released. The spores on falling on a suitable substratum will germinate, forming the thallus of the Marchantia. So, this is the Marchantia sporophyte, and this is the life history of full life history of the Marchantia. One is the thallus of the Marchantia. From the thallus of the Marchantia, this reproductive organs, like here this is the sporophytic phase, and this one is the archegonium, here the antheridium, from them the capsule, the antheridia, archegonia, then egg cell, fusion of the egg cell with the sperm or the antherozoid, then it converted to sporophyte, the zygote from sporophyte, and from the sporophyte it developed into a new gametophytic plant body, that is the thalloid plant body. So, in the life history of the Marchantia, the gametophytic phase is dominant because it takes place at longer duration, and the sporophytic phase is not so flourished, as sporophytic phase is only represented in the zygotic phase, that is the 2N phase. So, here that the gametophytic generation and sporophytic generation alters with each other. So, gametophytic generation overs with the formation of gamete, that is the male and the female gamete, whereas the sporophytic generation starts from the zygotic phase and it overs with the reduction division or meiotic division of the zygote, resulting in the haploid nuclei or the haploid meiospores. These haploid meiospores under germination, they continuously divide. First they form the germ tube, then that green thalloid like body under continuous division develops into flat leaf like structure, and rosette like arrangement of the thallus. This is the rosette like arrangement of the thallus with multi-lobed structure, and after that the thallus bears the number of stalked bodies, and these stalked bodies are called as the reproductive stalked bodies, that is the antheridiophore and the archegoniophore. Here antheridiophore contains the antheridia and archegoniophore contains the archegonia. So, this is the entire life cycle of the Marchantia. Thank you.